So those of you who've been joining me in the lectures will know I have promised you a lecture supplement to help us catch up on some of the neuro content that got cut off at the end of one of our lecture theatre recordings, and also in response to a bunch of questions that I've been receiving at the ends of lectures about what exactly the slides and what exactly the images in the textbook or in the neuro papers themselves are telling us about the relationship between the brain and cognitive psychology. How does an understanding of neuroanatomy help us as cognitive psychologists to understand our subject better? We're going to start here with a bit of revision. And as you can see here, we have a cartoon of a neuron. So you can see that we have the cell body over here on the left, the soma, and surrounding the soma are the dendrites that receive messages from other cells. The long tail of the neuron is the axon, where once enough charge has been received from the dendrites, the neuron is able to fire an action potential all the way down that long wiggly axon and pass that signal along to the axon terminal buttons, at which point another neuron may be able to receive those signals through its dendrites. Along the axon, we can also see what's described in this picture as the myelin sheath. And this is a coating around the outside of the axon that allows the action potential to travel much faster. So I'm going to pause for a moment to stop by one of the godfathers of cellular neuroscience, Santiago Ramón y Cajal. So unlike the yellow cartoon we saw just before, this is a cortical neuron, and we can see it has a very different shape to the one that we saw before. So in the center, we can still see that there is a cell body surrounded by a host of dendrites with little spiny protrusions that allow them to receive the incoming signals from other brain cells and other neurons in particular. It's a little bit harder to identify them, but down the bottom here, we can see the axon, which in this case is branching. Now, we normally look at illustrations of neurons where the axon is much, much longer than the dendrites. And that is indeed the case here as well, but we're just not seeing the full extent of the axon for this neuron. That axon, if it were drawn to scale, would be several meters long, possibly tens, possibly even hundreds of meters, depending on how far its axon projection goes. Another thing that's not always obvious about the brain structure when we look at these simplified cartoons is that brain cells from different parts of the brain have quite different structures and quite different patterns of connectivity. And we're not going to spend too much time thinking about that today, but it's nice to just be reminded that neuronal structure and function can differ depending on where in the brain a neuron lives and what kind of role it plays. These differences in brain cell structure and brain cell function have been mapped out in great detail by Brodmann in the early 1900s. Each color on the cortical surface represents cells that under the microscope have a different structural organization or a different shape. You might also recognize the Brodmann map from visualizations of two very well-known parts of the brain, Broca's area and Wernicke's area. Now, while the cytoarchitecture can tell us about the surface features of those surface neurons, what do they look like, what might they be doing, we also have to think about the relationships between those regions. So Broca's and Wernicke's are known to be connected by a dense white matter tract known as the arcuate fasciculus. I'm about to show you a photo of a real brain that has been opened up so that you can see the different cell types. So this wonderful photograph shows you the shape of the arcuate fasciculus from the inside of that fiber bundle tract. And what you can see here is that the color of the tissue is very different to the color of the tissue in the wrinkly bits around the edges. So this is to remind us that when we think and talk about brain cells, and when we think and talk about neurons, we can differentiate in our minds the gray matter, which is the cell bodies, from the white matter, which is those axon connection bundles that are all myelinated, and it's the color of the myelin that we can see here as white. I have a second visualization down on the bottom right for you, which is a kind of brain scan that allows us to see the shape and structure of these fiber bundles without having to take a surgical approach. So if you would like to color along while you listen to this part of the lecture, feel free to download this coloring sheet from the link here. It's completely free, and learn about brain cells as you go. So first of all, just as we've seen earlier, we have a neuron. We can see the cell body up the top with that wonderful branching dendritic arbor where the neuron can detect activity from other neurons that are nearby and collect enough potential to fire an action potential all the way down their long axonal projection to their axon terminals at the other end, where they may be fortunate enough to pass those signals on to other dendrites of other neurons. And as we've just seen, the cell bodies have that slightly translucent color that is known as gray matter. We've talked a little bit about myelin already, and one of the things I think it's helpful to understand is that the myelin itself is actually a different kind of cell. So these are the oligodendrocytes, and the oligodendrocytes are very attracted to axons that are firing often. So when they detect axonal firing, their arms or processes will stretch out in that direction and begin to wrap themselves around the axon, and they will continue wrapping and wrapping and wrapping and wrapping until that bundle becomes a very, very thick, very, very tightly wound, fatty layer on the outside of the axon. And it's this active cellular process that generates the myelin in the brain across developmental time. And because it's an active cellular process, what we have to realize is that if an axon is not firing as often, then those oligodendrocytes will start to recede their myelin wrapping and can even send their processes in a different direction. So the wrappings of the oligodendrocytes create a thick outer layer that insulates the axon, allowing the action potential to travel a lot faster along the length of the axon. I always like to mention the microglia because they are the trash recyclers of the brain. I've already mentioned that any brain parts that are not in use might end up being recycled. So the microglia are involved in that active process. The other cell type that we can learn about is the astrocyte. And astrocytes play a special role in the brain where they actually have their processes reaching out to the capillaries in the brain. So the capillary is shown here on the right. And the capillaries are the tiniest ends of the blood vessels in the brain that help transport nutrients to different brain regions when the brain regions have been very active. So the astrocytes are involved in wrapping themselves around the capillaries and transferring nutrients from the bloodstream to those other brain cells. So it's important for us to remember that the capillaries are transporting oxygenated blood and other nutrients all around the brain constantly. But if a region of the brain has been very, very active, it will be very depleted, and those capillaries will permeate with more oxygen or oxygenated blood to replenish the neurons so that they may fire again in the future. So hopefully, having learned a bit about the characters in the brain and how they are all actively involved with one another, we can now move on to thinking about how we measure brain activity. And then we'll look at how those measurements are related to our subject of interest. The first method we're going to talk about today is single unit recording. This is the most direct kind of measurement you can make of brain function because it involves actually recording the changes in membrane potential in an individual neuron. Now, obviously, to get an electrode in to do a recording from an individual neuron is very difficult work. And obviously, you need access to the brain. So one of the complications of single unit recording is that it's not the kind of study that you can invite a bunch of undergraduates to take part in, because you would have to expose their brains. And that's a very risky procedure. Instead, the only time single unit recordings ever go ahead with humans is when someone might be scheduled for a surgery, some kind of a life-saving treatment. And prior to their surgical intervention, there might be a paradigm that they are invited to take part in. Sometimes that paradigm is directly involved in helping that patient have a better recovery afterwards. And sometimes there might be another research initiative going on at the same time that they're lucky enough to be involved in. So whenever you see a single unit recording, just be aware that this is a very special case in neuroscience if it's data coming from a human. The next method we're going to talk about is electroencephalography. So electrical potentials, encephalo, the brain, ography, making a graph. So this is also a measure of neuronal activity, but in this case, the electrode is on the outside of the skull. Now, this brings its own complications because obviously you're not just recording from one neuron, because the neurons are incredibly tiny. So you're recording from a giant population of neurons, and the voices of those neurons are somewhat muffled by the skull and the wet tissue surrounding the brain, and also even the skin on the outside. So when we conduct electroencephalography, what we do is we make recordings from multiple sites all over the outside of the head, and it might look something like this. And then using those individual recordings from individual electrodes, there are a bunch of different analytical techniques we can use. What I should mention is that these continuous recordings will include any kind of electrical activity that's detected at the skin. So this will include the normally occurring electrical activity that is generated by the human body and the human muscles, but it'll also include things like any oscillation in the electrical field nearby. So if you're near some power mains, there will be evidence of that oscillation in the alternating current in the electrical signal recorded at your skin. And there may even be external waves detected that might be the background radiation of the universe in there as well. But it is still recording in millisecond precision the changes in electrical potential at the skin, and some of those changes are indeed coming from the brain. One way we can use the recordings that we make in an EEG is to look for event-related potentials. So that is changes in the pattern of electrical potentials that we observe that are time-locked to events that have occurred for the participant. So here's an example of a situation where we are recording a continuous signal while a person is looking at some pictures on a computer screen and also hearing some words. So you'll see the continuous signal towards the bottom, and you'll see the stimuli just above. So we can see that they might be seeing pictures of fruit and hearing words like banana shortly afterwards. Now, what we're able to do is segment that continuous EEG into units of time that are time locked to the events that we're interested in. So in this case, we might be interested in how the electrical potentials change in the presence of an auditory stimulus. So we can look for the window of time around about when the word banana occurred the first time and the second time, and take those signals, combine them with every other instance of a similar acoustic event that occurred throughout the experiment. And across those different events, we can create an average. So the grand average of an event-related potential helps us to average out some of those sources of noise I was talking about earlier. So there will be brain signals in there that are just about keeping your heart beating. There'll be brain signals in there that are just about keeping you breathing, digesting your breakfast. And what we're really interested in is the portion of that signal that comes from populations of neurons that are coding for the auditory stimulus. So in this case, we can see a grand average that is time-locked to the onset of an audio stimulus. Another source of neurological data that we have about neuron function is what's going on when portions of the brain are no longer functioning the way that they used to. So in this case, a particular kind of disruption to neurological function comes in the form of lesions. So this is any instance that involves an injury to the brain where some part of the brain dies. This could be caused by a traumatic head injury of some kind, or more commonly, something like a stroke. What's interesting about brain lesions is that they can tell us something about which parts of the brain might be functionally involved in a particular cognitive function. So these are the brains of Broca's two first patients. Broca was one of the first to document that in his clinical work, when he observed patients who had particular kinds of language impairments, he was able to identify later on in post-mortem that they tended to share patterns of neurological damage as well. Now, clearly, lesion data is somewhat limited in cognitive psychology, as we can't really use it for experimental purposes. That is, we can't really damage someone's brain and just see what happens at the other as an outcome of our interesting experiment. Fortunately, we do have some techniques in contemporary neuroscience that help us approximate this kind of causal relationship. So the method of transcranial magnetic stimulation, also known as TMS, allows a researcher to temporarily disrupt neurological function. So in the method of transcranial magnetic stimulation, the way it works is we have a coil like the one you can see in the center of the screen. And as you may remember from your high school science class, every time you pass a current through a coil, it generates a magnetic field that rotates around the center of the coil. Now, if it's a continuous current, that magnetic field will go in one continuous direction. If it's an alternating current, then the magnetic field will oscillate in and out of the center of that coil. So obviously, as we've just observed, we know that the firings of action potentials are an electrochemical process. So if we introduce a strong magnetic field into a location where axonal firing is normally occurring, it can disrupt that process temporarily. But the moment we remove that magnetic field, then function should resume as usual. Most contemporary TMS uses a technique with two coils. So that means that the magnetic fields are overlapping with each other somewhat. And that area of overlap has a much higher concentration of magnetic field than is present for the surrounding tissue. So this allows the method of transcranial magnetic stimulation to be highly targeted to particular brain regions. And it's also entirely reversible. So you can just switch the machine off. So this kind of technique allows us to test a very strong hypothesis that a particular brain region is involved in a particular function, because we can test whether or not a person can carry out that function and then cause a temporary impairment in that region and see whether the function can no longer go ahead as usual. The next method that we're going to hear a bit about, and the one that you're probably feeling quite familiar with, is MRI, magnetic resonance imaging. So MRI is a highly spatially accurate scan, and it can be used in a variety of different ways, because the giant loop that you can see in this image contains a very, very high-powered electromagnet. And the field intensity inside that electromagnet can be changed in a variety of ways, different spin cycles, to generate different kinds of measurements from the tissue that is inside the scanner. It measures the position and density of radio frequency absorbing molecules. So in a way, it has a great sensitivity to certain kinds of tissue that contain a lot of water and certain kinds of tissue that contain a lot of fats. Now, since cell bodies are very watery, the gray matter and axon myelination results in a very dense fatty layer around the outside of the axon. That means that the white matter is going to be detected as structurally different from the gray matter. So every time you see a scan that looks like this, where you have an outline of a skull, and you can see a brain inside, and it's wrinkly on the outside, and you can see darker and lighter portions inside the brain structure, you're seeing those cell bodies and you're seeing those dense fiber tracts, the gray matter and the white matter of the brain. There are a couple of other ways that MRIs can be used. And again, this is just a different scan cycle. And one of the really interesting ones is diffusion tensor imaging, also known as tractography. So this is a way of using the electromagnet inside the MRI to map out the ways that water can move within the brain structure. So every water molecule in the brain might be moving around a little bit as it bumps into other molecules inside the wet soup that is the human head. And as they do so, some molecules will be more constrained in the directions that they can move. So those water molecules that are outside cell bodies will be able to move in all directions quite freely. Those water molecules that are on the inside of an axon will be very limited in the direction that they can travel. They can travel up the axon, they can travel down the axon, but they can't really travel directly through the cell membrane. So tractography is a technique that allows insight into the unseeable parts of the superhighways that connect different brain regions. Now, functional MRI is different again. In functional MRI, the scan cycle is controlled in a way that allows it to detect the amount of oxygenated blood in different parts of the brain. So we talked earlier about how the capillaries in the brain are bringing oxygenated blood to different brain regions. And it's particularly important when one brain region has been very active, that those capillaries draw in more oxygenated blood to replenish those neurons so that they're able to continue firing. So this is oxygenated hemoglobin that can be measured by the bold signal in an MRI. What's important to realize, though, is unlike some of the electrical signal measurements we saw earlier, and unlike the TMS disruption of electrical signals, we're not actually measuring anything to do with the electrical activity of the neurons themselves. This is an ancillary process that goes on in the brain that is logically linked to the activation of neurons. It is important for us to realize, though, that it's a blood flow process that happens after the firing has occurred, and it also functions on a different time scale. So MRI in its functional mode, functional MRI, has about a 1 second window where when an activity has occurred, it takes about a second before it's detected in the bold response. Now, because you'll be seeing lots of images of brains, it's helpful for us to break down what it is you're going to be looking at. So here we have just a little explainer for what is an MRI slice. So on the left-hand side, you can see a cut through of the side of the head, and I've drawn a little blue outline so that you can kind of imagine where that would be on a person. The other thing that we can see on this left-hand image is that a few green lines. Now, those green lines are indicating where a measurement slice has been taken. The imaging has been analyzed as though it's just a single sheet of data from each of those green lines that you can see. In the second image, the image has been rotated and we're now viewing from the front. And you can tell because you can also see those big black orbs where the eyeballs are. So in the second image below, image B, we have exactly the same thing again. This is the same scan cycle, but this time those green lines are a little bit further back in the head. So instead of being up here where they cross over with the eyeballs, they're now back around about the midline. Because it's further back, we can see that the two head shapes are different. And the shape of the brain within the cranium is different. So whenever you see different size, different shape pieces in one MRI image, it's important to realize that they might be from the same moment in time, just from a different position on the head. This is the final kind of brain measurement we're going to think about today. And it's another way of utilizing the data that has been collected during functional MRI. What we might do is say, why don't I correlate the function of that first box on the top left with every other box in the brain and see which of the other boxes are most strongly correlated with it. And then I'll move to the next box and correlate it with everything except the first box. And then I'll go to the third box and do the same thing again. And then the fourth box and the same thing again. And this is the kind of data pattern you might expect to get from this. Now, in my example, the boxes are gigantic. When this is done for real, the boxes can be as small as 1 millimeter by 1 millimeter by 1 millimeter. So a cubic pixel worth of brain, a voxel. And when you think about the number of voxels you have in the brain, there are just far too many correlations for this to be a worthwhile approach to brain connectivity. So this approach seems like it's not going to get us far because some of those correlations are going to be significant just by chance, just by random chance. The second option that we have is we can pick a particular location of interest and use that as the starting point for all of our correlations. So if we were to pick box 6 in the brain and say, how is box 6 related to every other place in the brain? We would be targeting a specific region of interest and finding out which other regions are most strongly co-active. And it might look something like this, where we set a seed in box number 6 because we know of a particular function that's very important for us there. And then we can track the pattern of correlations through the rest of the brain to try and find out how activity in the first location is related to activity in other locations. Okay, so that brings us to the end of our summary on neurons, brain cells, and scan types. I hope that this primer has been a nice intro to a bunch of different brain scanning technologies and a bunch of different ways of understanding the way that the brain works.